early life stress modulates how this response to LPS is. Early life stress by itself increases some of these cytokines, for example, IL-6 and CXCL1 and CCL2. But also there is an interaction. So this early life stress aggravates some of these responses in cytokines in response to LPS. This was quite a harsh LPS, I must say. In hindsight, we should have probably could have been better using a little bit a milder LPS injection. This is also why we have such a plateau in LPS response. But nonetheless, we see some interesting effects. So what happens to gene expression analysis? Does the diet impact on it under basal condition? Does it affect on it in response to LPS? And is this dependent on early life stress in any possible way? So I will just show you a little bit of this data, of course. So first of all, we see that an early diet, which is long gone, impacts on the gene expression profile of the hippocampus. And what we see, for example, we see, of course, many different geoterms, but we see, for example, indeed changes in long-chain fatty acid transport and long-chain fatty acid catabolic processes, kind of confirming that this diet indeed somehow might be incorporated in the hippocampus. So this is just a comparison between controls and under the two different diets. And what you see here with these Venn diagrams is that you can compare, of course, the comparisons. So you have the controls and early life stress under the high diet, so with a certain ratio of fatty acids, and the controls and the early life stress under the low diet. And what is striking to see is that indeed, in both cases, there is, of course, altered gene expressions, but that these are almost entirely different. So depending on the diet, we have an entirely different set of genes that are being altered by early life stress. One remarkable one that will come back also later is, for example, that under the low diet, which is the one that actually rescued our early life stress-induced phenotypes, is the extracellular matrix, which is one of the top hits. What happens in response to LPS? So LPS induces also gene expression changes that are different in controls and early life stress mice and depend on the diet, of course. So what we see again is that we have a completely different set of genes depending on the diet and depending on early life stress and on the LPS. And again, even in response to LPS, what we see with the arrow is that under this low diet, in response to LPS, the LS and ELS animals exhibit changes in relationship to extracellular matrix, which might be then an interesting target to approach. So what happens with the hypothalamic profile, lipid profile? So this is also something that we tackled in this study with a really similar comparisons. And I will just give you a glimpse of that. What we see is that this diet that was given early in life, many months later, actually alters the lipid profile that you find back in the brain, which is maybe also to be expected, but still it was really interesting to see it categorized as such. And what we see is that it decreases hypothalamic lipid species implicated in inflammation and stress. We also see that this low diet ratio differently modulates the lipids in controls and early life stress animals. So again, we see that there are not that many lipids changed, but the ones that are changed are different depending on the early diet. And last but not least, what is really striking and interesting is that LPS, just in response to LPS, we actually alter our lipid profile in the hypothalamus, and this is highly dependent on early life stress and on the dietary ratio. What you can see here with this more complicated Venn diagram. So I think in summary for now, we see from the hippocampal gene expression that this is affected by early life stress, of course, by dietary ratio, under basal level, and also in an immune challenge condition. And that may be one of the hits for how these beneficial effects of the diet is, which was actually the question that we had to begin with, might be related to the effect that it has on extracellular matrix and transmembrane receptors. So this is something that we are currently following up. Similarly, the lipid profile was affected by all of these different conditions. So what we are currently doing is actually studying further the gene expression analysis, also with the microRNA data set, together in collaboration with Dr. Cattaneo from Milan. And we are also looking at not only at the lipid profile, but also the SPM profile, so the specialized pro-resolving mediators, to understand even further how the signaling aspects of these fatty acids are. So just a glimpse of what we do with the micronutrients, because that was also part of the goal. But the micronutrients that we focus on are the ones that are belonging to the one-carbon metabolism. So they are really important for methylations, for producing methyl groups that are important for DNA methylation and for histomethylations. And these are, for example, folic acid, vitamin B6, B12, etc. So again, in a study of a few years ago, we have shown that an early supplementation with these micronutrients is able to to rescue some of these phenotypes of the early life stress. First of all, early life stress changed the methionine composition of the brain, it reduced it. And what we see is that with the diet, we managed to rescue these methionine levels, we managed to rescue the cognitive deficits, and what was really interesting and striking to us is that the HPA axis seemed to be modulated by this. So while we have normally an increased corticosterone in response to this early life stress, this was completely absent in the presence of this micronutrient. So this led to our next question, which we did then in collaboration with Veronica Begni from Milan and Kitty Reimst. We supplemented the same diet as before, but instead of waiting to adulthood, we looked in adolescence, and instead of only looking at basal level, we also looked in response to acute stress, again with a similar principle as with the LPS earlier. So it's a quite complicated study again with many groups, and we submitted the animals to a long battery of tests to really tease apart the anxiety phenotype of this model. We exposed them to elevated plasmase, the dark light box, the splash test, and sucrose preference test at the end. And what we see basically is that indeed the early life stress leads to changes which are often apparent only in response to acute stress. In this anxiety phenotype, so elevated plasmase and dark light box by early life stress are mostly affected in response to acute stress, and this is rescued by the diet. The splash test was a little bit more obvious already at basal level, but also in response to acute stress. And again, this effect was modulated by the diet. We did not see any effect on the sucrose preference, but this was also quite far from the acute stress exposure itself. So we think that that's actually the reason for that. What we did next, we wanted to see, okay, our main goal with these micronutrients was actually to modulate, hopefully, possibly, the epigenetic machinery. So in collaboration with Moshe Sif, we went and we did a genome-wide methylation analysis in the hypothalamus to understand what are the effects there. And what we found was actually very interesting. First of all, most of the sites, 86% of the sites are hypomethylated by acute stress in control animals alone. This is what you see in these first two bars. Early life stress alters the methylation levels upon acute stress. This is what you see in this comparison. So this is really, really interesting. And many of these sites actually are on sex chromosomes, on the X and Y chromosomes, also implicated in neurodevelopment. And last but not least, what we were also, of course, really happy with is that the diet reversed most of these effects that were induced by early life stress. So the animals that have received the macronutrient early in life, during the adolescence, exhibit a very different methylation profile. We, of course, looked at the relationship of these methylated sites with the behavior, and there were some that were also correlating, 74 the differential of these different sites also correlated with behavior. So I think I will skip the summary because I'm already a little bit over time. One last slide that I would like to show you is that, of course, we are also intrigued by where does this different nutrient composition comes from upon the stress. And one of the hypotheses that we are pursuing is the possibility that the stress exposure of these dams affects the maternal milk composition. And we are doing that actually in a human study. And we have the first results on the fact that stress indeed correlates with the altered fatty acid composition of human milk. And in addition, bringing back also to the presentation of Suzanne, we also have found that early life stress affects actually food choice later in life. So it's possible that another additive value is also the type of choices that an early life stress individual makes later in life for what they eat and what they don't eat. So just to wrap it up, I would like to one more highlight that animal models of early life stress are translationally very valuable, that next to stress hormones and sensory stimuli, which have received a lot of attention in this context, nutrients play a key role, that nutrition interventions are very promising and could be a new potential avenue, but we need to learn more. Nutrients could also serve as a contributing factor to the search of the biomarkers for vulnerable individuals. So we need to understand further about the mechanism and how all of this interacts in order to be able to optimize the current nutritional strategies. I thank you for your attention, and of course I thank all the collaborators and everybody that did the work. Thank you.
Thank you very much. And we go on now with, I guess, going to a different animal model. I think this will be very interesting to see how these mechanisms that we have seen so far can be applied in an animal model like Drosophila. And this will be, the speaker will be Carlos Ribeiro. He's actually group leader here in Lisbon at Champalimud. And his talk is the PLAP, nutritional decisions in Drosophila, including the impact of the gut microbes. Thank you.
Okay, thank you very much for the kind introduction. It's also great to be here finally in an in-person meeting and not staring at a screen. So today I'm going to, I think, give a bit of a more exotic talk for this setting and tell you about our work in Drosophila and trying to understand how brain-body interactions shape nutritional decisions in the animal. So I would like to start by thanking my team. Oops, okay, no, this is not, okay, the mouse is really not working. So I would like to thank my team, a fantastic group of people who have, whose honor I have to be the spokesperson. Also thank our collaborators. I think working on nutrition and the brain requires a lot of interdisciplinary work, and it's really thanks to our collaborators that we can really span work from behavior to metabolomics to genomics and many other things. And then finally also our funders who keep us and our flies fed. And here I would really like to emphasize and thank the Champalimaud Foundation. This is our center. It's the Champalimaud Center for the Unknown. It's a state-of-the-art cancer and brain science institute, which if you walk here along the river towards the ocean for 5 kilometers, you can go there, and I think on Monday there's going to be a very nice ALBA reception there, so I encourage you to go there. So if you think about what most neuroscientists are interested in, or most of the work actually has been done in neuroscience about food, is the question of how do animals decide to eat or not to eat. And I think while this is a very interesting and important and extremely productive field, I think if you think about it, what the animal really needs to solve is the problem of which nutrients to eat in which amount, right? Because every different nutrient has a very different impact on health and behavior. Energy has a very different impact than vitamins, and proteins and fats also have different impacts. And so how to really reconstitute a complete and optimal diet from these different types of opportunities is something which I'm really interested in. And we're focusing our work mostly on amino acids and proteins, right? So if you think about it, amino acids are obviously essential, right? We cannot make proteins without amino acids. And in contrast to other nutrients, we cannot make 10 out of the 20 amino acids, so they're essential. And they are actually more or less the same essential amino acids in all animals, including Drosophila. And so that means that we have to actually eat them to be sure that we have enough of them. But interestingly, actually, just overeating amino acids so that you're sure that you always have enough amino acids is also not an optimal strategy. Because I think work over the last 20 years has shown in almost all animals ever studied, and including epidemiological studies in humans suggest the same, is that overeating amino acids and proteins shortens lifespans, as has a very negative impact also on health span, right? So we need to eat enough amino acids, but we cannot overeat it because that will shorten our lifespan. And so we want to understand how the brain deals with this trade-off and solves it. Now, thinking a bit more translationally, I think there's also a lot of interesting evidence that amino acids have a very strong impact in neuropathologies in the brain. And here I want to just highlight one study from my colleague Gaia Novarino, who's now at IST in Vienna, which has shown that in certain families where children have a very severe cause of autism spectrum disorders, there are mutations in the genome which all seem to impair branched-chain amino acid uptake and metabolism in the brain. So there seems to be a link between amino acids